Hello, I'm Igor Busato. I'll talk about this work, which I am a co-author, and it is entitled as Evaluation of the Effects of Chronic Exposure to the Herbicide Monosodium Methyl Arsenate MSMA on the Epididymis morphology of Wistar rats. It is well known that the agriculture in Brazil is very dependent on the use of pesticides. In 2009, the country reached the position of the world's largest consumer of this product. One of the herbicides widely used in the country is monosodium methyl arsenate, MSMA. Despite this, MSMA was classified by IBAMA as very dangerous due to its potential environmental danger. The threatening regarding MSMA is associated with its composition, which is derived from the heavy metal arsenic. Arsenic was associated with oxidative stress and changes in the enzyme's function, also attributed with compromised seminal quality and reduced weight of accessory sexual organs, such as the epididymis. The epididymis is responsible for the maturation of sperm, making them mobile cells and capable of recognizing and fertilizing the oocyte. Besides that, the epididymis also plays an important role in the transport, concentration, protection, and storage of sperm. Thus, agents that affect the morphology of this organ can cause infertility. Thus, considering the toxic potential of MSMA, this study analyzed the effects of chronic exposure on the morphology of the epididymis and sperm of Wistar rats. In this study, 16 rats on reproductive age were separated into two groups. First, the control, which was treated with water, and the treated group, which was given 120 mg killer per day of MSMA. The treatment lasted 60 days, and the experiment was approved by the Ethical Committee of Universidade Federal do Espírito Santo. Then, 24 hours after the end of the treatment, the animals were sacrificed and their organs collected and processed for light microscopy and for other analysis. Regarding the analysis, we weighted the rats at the beginning and at the end of the treatment, we weighed the epididymis, we did histopathology of the epididymis, and we analyzed the sperm morphology, DSP, sperm reserve, and sperm transit time. After that, the results underwent statistical analysis by students' t-test. In regard to biometrics, the MSMA treated group had a lower final body weight and also a lower weight gain compared to the control group. In addition, the epididymis of the MSMA group had lower weight compared to the control group. Other studies point to similar results, and knowing that body weight is a measure of toxicity, these values found may indicate toxicity and possible injuries generated by this arsenic-based compound. Previous studies by the research group with acute exposure to MSMA showed no reduction in organ weight. On the other hand, this chronic exposure seems to reduce the mass, which may corroborate our group's hypothesis that the harmful effects of arsenic in the body are time-dependent. Histopathology indicated that the epididymis of rats treated with MSMA have a tissue architecture similar to the control group, however, with some mild alteration. In the intertubular compartments of the MSMA treated group, an increase of macrophages proportions was observed. According to the literature, the increase in these cells may be associated with a response to oxidative stress induced by arsenic. In the tubular compartment, rats treated with MSMA showed microvacuolizations in the epithelium and abnormalities in the lumen of the epithelium, such as accumulation of cellular debris and reduced amounts of sperm and a greater presence of abnormal sperm. The literature indicates that the vacuolization of the epididymal epithelium is the result of degenerative changes in which there are intracellular accumulation of fluids and other cellular components. Regarding the luminal changes, the expressive presence of germ cells in the tail lumen of the treated rat epididymis may have been the result of lesions in the testicular germinal epithelium, which are caused, for example, by the androgenic decrease generated by the action of toxic agent. Thus, the increase of this debris in animals exposed to MSMA may indicate degenerative processes in the testes of these animals, triggered by the toxic effect of this compound. The values of DSP, sperm reserve, and sperm transit time were lower in the treated group compared to the control group. It can be inferred that the reduction of the sperm production rate and the DSP may reflect damage to cells that are part of the germinal epithelium due to the oxidative stress in the treated group. The shorter sperm transit time in the head, body, and tail regions of the epididymis of the rat exposed to MSMA may be related to problems in sperm maturations. Although some authors indicate that the reduction in transit time is the cause of defects in sperm, its increase can also cause damage, since the prolonged permanence of sperm in the epididymis can make them susceptible to oxidative stress or external agents. Finally, sperm morphology analysis indicated a higher proportion of abnormal sperm in the MSMA treated group compared to the control group. Here we can observe the abnormalities found: sperm with normal head but with the flagellum separated, and sperm with the hook at a different angle. These damages to sperm morphology may be associated with the increase in transit time observed in this study, since the long permanence of sperm in the epididymis can cause changes in several ways. The literature also associates these morphological abnormalities in sperm with reactive oxygen species generated by arsenic, negatively impacting the overall quality of the sperm. In conclusion, MSMA needs more attention and consequently more studies to evaluate its effects on the reproductive system and fertility.